Assalamu alaikum. I am Dr. Asma Niazi and I welcome you to the fantastic world of microbiology. Today we are going to study one of the antibiotics, which is tetracyclines, and which is one of the protein inhibitors. The sequence in which I will teach you about tetracycline is as shown on the slide. So the tetracyclines were discovered in 1948 by Benjamin and M. Duggar from a fungus which is Streptomyces aureofaciens. The newer antibiotic of tetracycline, which is tigecycline, was discovered in 2005, and further newer agents were developed in 2018, and that include eravacycline, sarecycline, and omedacycline. Tetracycline molecules comprise of linear fused tetracyclic nucleus designated as A, B, C, D as shown on the slide, to which a variety of functional groups are attached. While tetracyclines have common structure, they differ from each other by the presence of chloride, methyl, and hydroxyl groups. The tetracyclines are classified in two ways. One, which is shown, it is according to the half-life of the tetracyclines. These are divided into short-acting, intermediate-acting, and long-acting. The short-acting have a half-life of 6 to 8 hours and include tetracycline and oxytetracycline, whereas the intermediate-acting have a half-life of 12 hours, which include demeclocycline and methacycline. And furthermore, there are long-acting that have a half-life of 16 hours, and that include doxycycline and minocycline. So the second way to classify the tetracyclines is according to their generations, यानी जो पहले बनी थी और जो अब नई बनी है. So in the first generation, the tetracyclines, the chlortetracyclines, the oxytetracycline, and the demeclocycline and metacycline are included, whereas in the second generation, we have the doxycycline and minocycline. The third generation, as I told you before, include digicycline, which was discovered and in 2004, whereas the newer agents that were made in 2018 were eravacycline, sarecycline, and omedacycline. The mechanism of action of tetracycline is the tetracyclines enter the bacterial cell wall in two ways: passive diffusion and an energy-dependent active transport system. The tetracyclines work by inhibiting bacterial protein synthesis. This is accomplished primarily by reversibly binding to 30S ribosomal subunit of the bacteria. As is shown on the slide, that there is a tetracycline binding site. This inhibits the binding of the amino acyl transfer RNA to the adjacent ribosomal acceptor site, which in turn prevents peptide chain elongation and inhibits protein synthesis. Since the binding with 30S is reversible, it is postulated that this is the explanation as to why they exhibit bacteriostatic properties. The tetracycline had previously been widely used for an array of respiratory, gastrointestinal, and genitourinary tract infections. The widespread increase in resistance, along with more and newer bactericidal drugs, has decreased the number of diseases where they are now considered to be the drug of choice. These diseases include, for which they are used for the drug of choice, are chlamydial infections, rickettsial infections, in Lyme disease, brucella, infective exacerbation of COPD, granuloma inguinale, chancroid, relapsing fever, and tularemia. The clinical indications for individual antibiotics are: for tetracycline, it is used for rickettsia, chlamydia, mycoplasma, alternative therapy in syphilis, gonorrhea, part of combination therapy of Helicobacter pylori, and tularemia. The doxycycline is used as for Borrelia brucellosis, Yersinia pestis, Francisella tularensis, Vibrio cholerae, Urea plasma urinalyticum, Q fever, Lyme disease, inhalational anthrax, and community-acquired MRSA, methicillin-resistant Staphylococcus aureus. Other major uses are in rickettsia, chlamydia, and mycoplasma. The minocycline is very much used in acne vulgaris, Lyme disease, and most active against MDR, multi-drug resistant, gram-negative bacilli, for example, Acinetobacter species and Cynotrophomonas. Other tetracyclines like omedacycline is used in community-acquired bacterial pneumonia, acute bacterial skin infections, whereas sericycline, moderate to severe acne vulgaris, and eravacycline is used in a complicated intra-abdominal infections like appendicitis, cholecystitis, peritonitis, and gastric perforation. These are the new generations of the tetracyclines. To help you in understanding the tetracyclines and their diseases associated with tetracyclines, I have made a mnemonic. The word tetracycline can be used for the first word is used for the number of diseases that it is used for the treatment. Like T for tick-borne diseases, E for exacerbation of COPD, T for tularemia, R for rickettsia, A for atypical pneumonia, C for cholera, Y for Yersinia, C for chlamydia, LGV, Lyme disease, both of diseases, inguinale, that is the granuloma inguinale, Neisseria, alternate treatment के तौर पे use होता है, and the epidemics of plague. This can be easily remembered. The adverse effects include those of the gastrointestinal tract are nausea, vomiting, anorexia and diarrhea, anal pruritus, vaginal or oral candidiasis, and enterocolitis. As the picture is shown, it also has some adverse effects on bones and teeth like discoloration and enamel dysplasia. Moreover, it also causes bone deformity or growth inhibition. Further, on the liver, it causes impaired hepatic functions and hepatic necrosis. In kidney, it causes renal tubular acidosis, while hemolytic anemia, like thrombocytopenia, neutropenia, and eosinophilia are some of the hematological adverse effects of tetracyclines. I have taken this topic and its multiple aspects from from these books, as I have showed in the references. I am very thankful to you for your patient listening. Thank you.